In this lecture that is part of the series on fMRI data analysis, we'll be looking at the fMRI paradigm design. Here's an example of a typical fMRI experiment that I've shown a number of times. This is showing the activation in the bilateral motor cortex occurring during bilateral finger tapping. One important thing to keep in mind about functional MRI is that we are measuring relative changes in the brain activity. That is, we can't measure the amount of activity directly, rather we have to create some sort of paradigm where we're looking at the changes in activity relative to some control condition. So in this particular case, we had the participant tapping their fingers on both their hands for 20 seconds, alternated with 20 seconds of rest, and so on. So one question you might ask is, why are we performing the task in this manner? That is, these blocks of task for tens of seconds alternated with resting blocks. Well, perhaps a much more simpler design that you could think of is this one illustrated here, where we collect some images during a control condition, and then we have the person engaging on some sort of task, and we keep collecting the task, keep collecting images, and then we look for where do we see differences during the control or during these task periods. The challenge with this kind of design is that the fMRI signal also has very slow drifts in it. And in that case, a very slow drift or signal increase might look like this task activation response. So an alternative might be, you could say, well, let's have the control condition at the beginning and at the end to control for a slow linear drift. And indeed, some of the early fMRI experiments were done this way. Here's an example from Xiaoping Hu showing visual activation during watching some flashing lights from visual stimulation. And in this particular case, he had a period of time when the person is resting, then watching the flashing lights, and then resting again. However, this was acquired at a very short repetition time of 0.3 seconds. So this is about 30 seconds of rest, 30 seconds of stimulation, and then 30 seconds of rest again. If we're dealing with much longer time courses, again, if these signal changes are very slow, it might be very difficult to distinguish them from any kind of slow drifts. So because of this, we acquired the task much more rapidly, periods of on the order of tens of seconds. Now, this is a set of experiments that we call a block design stimulus, where we have the task together in blocks. Another kind of design is what we call event-related designs. That is, we think of the tasks or the stimuli as individual events. So the question then becomes is, what is the optimal timing of this kind of a design? So we can look at this. This is some results from a simulation. And what I'll be showing you here is the example response and how it varies with different stimulus onset asynchronies. That is the time from the beginning of one stimulus to beginning of the next stimulus. And as to here, the stimulus duration I've fixed at 1 second. So again, if we have a brief event or brief task happening every 32 seconds, this is what the response might look like. If instead I have the brief response or the brief task happening every 8 seconds, this is what the response looked like. And you can see that it barely returns back to baseline. If I have the task performed every 4 seconds, you can see that the signal increases and just sort of varies around a little bit. And now if I perform the task with an SOA of 2 seconds, that is 1 second task, 1 second rest, 1 second task, 1 second rest, you can see that the stimulus, that the signal really just increases and doesn't really vary much at all. So here we've really lost our ability to detect this kind of a response, aside from this initial increase. And this was actually demonstrated experimentally. Here's an earlier study from Peter Bandittini. And you can see this is an example. In this case, now the inner stimulus interval is the rest period, and this is the stimulus duration. So for 20 seconds of rest and 20 seconds of stimulation, we saw a nice robust block design response. If we have 2 seconds of task, in this case, motor cortex finger tapping task, 12 seconds of rest, here you can see the response. 10 seconds of rest, you can still see a nice response. But as that inner stimulus interval gets shorter and shorter, eventually we reach a case where the signal increases and really doesn't vary around very much. And we lose our ability to detect this kind of response. So based on this, you could come up with some general formula for what the ideal interstimulus interval or total period should be based on the stimulus duration. Now, what's actually interesting is that the same year that this was being presented at the ISMRM conference, another study showed that you can actually get very robust responses, even from very short interstimulus intervals. And the trick was to vary the interstimulus interval. So at the top, you can see what happens with the fixed interstimulus interval or intertrial interval of 16 seconds. You see the nice response. 3 seconds, really don't see very much variation at all. And a 1 second intertrial interval of rest period, we really don't see any variation anymore. However, if you take the same number of stimuli and just vary the timing between them, that is, you jitter the intertrial interval, you can recover a lot of the response that you would otherwise have lost. These event-related fMRI designs have a number of advantages. One is this event-related designs might be more appropriate to the task. For example, if you're interested in studying swallowing, that is something that is an individual event, not very easy to perform in a block. In addition, you can make the task more unpredictable, which allows for less neural adaptation. This is very advantageous if you're trying to understand, for example, memory or inhibition using, for example, a go, no-go task where the no-go event happens at some undetermined amount of time later. And for example, also to maintain the participants' attention levels. Event-related designs can also be used to sort the events based on the participant's response. So for example, you can look at what's different about those stimuli that were later remembered versus those that were forgotten, or the correct versus incorrect trials. And you can see what happened during the activation during these two types of periods. In addition, these event-related designs can also be made less sensitive to task-induced motion, such as speaking and swallowing. A really good, perhaps old reference about these event-related designs is a PNAS paper from Bruce Rosen from 1998, and I'd highly recommend you looking at this if you're interested in this further. The question then becomes is how do you actually design or find the best stimulus pattern given all of these different variables? And for this, I draw your attention to one of two possible analysis packages. One, the AFNI analysis package, where you can use the 3D deconvolve. This is the main program to do multiple linear regression. You can use that with the -no data option in order to compare different kind of task designs, and you can generate different kinds of task designs using either RSFGen or a Python script called make random timing. In addition, there's a script written by Doug Grieve called optseek from the NGH group. In addition, I have another lecture that you're welcome to check out that goes into some of the rule of thumbs about pair design in greater detail. Here's another kind of fMRI paradigm, in this case used for retinotopic mapping. If you look at how the visual cortex is organized, things that are in the center of your field of view generally appear at the most posterior part of the visual cortex, whereas things out in the periphery move more anterior. In addition, it is also organized such that different polar angles of what you see in your visual field also have a nice retinotopic organization. So one way that this was mapped is by showing a particular stimulus, in this case, a contrast reversing checkerboard. That is basically, initially, you can show two types of stimuli. First, you can show this ring of a contrast reversing checkerboard slowly expanding, and then going back to the center and slowly expanding again. And by looking at the phase, that is the timing of the signal changes, you can find out at what time different parts of the visual cortex were activated, corresponding with when they were stimulated. That is when the flashing checkerboard was in that part of the visual field. So essentially looking at the phase of that particular signal. And similarly, you can look at the phase or the timing of the signal as it's rotating around to determine where in this polar angle the visual activation is occurring. And using this, you can actually map the visual cortex in different areas of the visual cortex. One interesting variation of this retinotopic mapping is what has become known as Buffy-o-topy. This is another study where they did a very similar experiment, but they actually showed Buffy the Vampire Slayer, a TV show, in the background, but only showed a small segment of this, either in terms of a ring that's expanding or a small wedge that was rotating around. And by doing a very similar kind of analysis, you can map out the different visual areas. This actually proved to be quite good at mapping out the visual areas because the participants were quite engaged in the task because they were very interested in seeing what was going on in the background, all of these different changing characters and changing intensities there that still activated the visual cortex. But because the attention levels were so high, it actually boosted the activation that you saw. And finally, another set of tasks that you could do is what we call naturalistic stimuli, that is just watching movies. So you could show a particular movie to all the different participants, and you could try painstakingly go through and tag different parts of the movie as well. There was sounds during this particular scene and maybe faces on the screen during this particular scene and code them that way. But another way to analyze that data is to look at the commonalities in the activation across participants. So for example, if you showed the same movie to 5 different subjects, then at a certain point in time, all the different subjects would be seeing a face on the screen or would be hearing some sort of sounds, and therefore their activation should be correlated. So by looking at the correlation of the activations across subjects, you can actually find these interesting maps and map out these different functional areas. So hopefully I've given you some idea of the different kinds of paradigms that are possible with functional MRI. The main thing to remember is that with fMRI, really looking at the changes in activity, and that is really, I think, what is the most challenging about designing fMRI studies, is to try to find out what the proper control conditions are to really isolate what you're interested in.